The resolving power of modern electron microscopes is truly stunning. And let me show this movie to illustrate it. In this movie, we took gold beads and placed them on a carbon film, and then we imaged them in a high-powered electron microscope and just recorded the images. And so here you see the gold beads. They're roughly spherical, and as the electrons are hitting them, the electrons deposit energy, which causes the gold beads to roll and tumble a little bit. And while they're rolling and tumbling, you see every once in a while a set of distinct planes through those particles, and you can see between those planes. And what those planes are is planes of gold atoms in the gold lattice. And this is just meant to illustrate how modern electron microscopes can easily resolve the distances between atoms. In fact, in some material science applications, electron microscopes have been used even to learn a little bit more about the shapes of electron orbitals surrounding particular atoms. Now, as cell biologists, as we watch that movie, we wish we could apply that same resolving power to cells and really see what's going on inside. But as we do so, we encounter three fundamental challenges in biological electron microscopy. The first challenge is, how do we preserve the native structure of a cell within the ultra-high vacuum of the electron microscope? I mean, you can't just swab some cells out of your cheek or in a tissue culture plate and then immediately put them into the column of the electron microscope. Because the vacuum is so high, all the liquid would immediately boil so explosively that all of your cell structure would be destroyed. So how do we preserve the structure of a cell and have it in the electron microscope? The next challenge is, how do we obtain three-dimensional information from projections? We may have a cell, but the electron microscope delivers a projection through it. So how are we going to obtain the three-dimensional images that we need? And finally, how do we overcome the dose limitations? The problem here is that the high-energy electrons that we're using to image the cell break the covalent bonds in the molecules of the cell. And if you have two atoms that are covalently bonded, they're close together. And if an imaging electron breaks that bond, then those atoms push apart. And as they push apart, if this is happening to many of the atoms of the molecules, they will all push apart and you'll get damaged material. You won't be able to see the native structure anymore because it's gone.